Thank you very much, Fritz, for agreeing to give a Build a Cell seminar, and we can get started.
Okay, thanks, Kate. Thank you very much for inviting me. It's exciting for me to give a talk in your seminar series, which I should attend also regularly. I feel guilty, but my name is Friedrich Simmel from TU Munich. I'm based in Munich. I'm a physicist. I'm working in biophysics on various topics. So the main topics of my group are essentially DNA nanotechnology. So that's maybe the more some more molecular kind of direction. But then we were also interested a lot in making synthetic gene circuits, and these we investigated over the last years, both in a cell-free context, but also more in a standard synthetic biology context within cells. And of course, we were also interested in compartmentalizing these systems, which sort of connects the research to probably creating synthetic cell-like systems. Today, I wanted to talk essentially about two topics which are related to cell-free gene circuits and maybe not so much to really creating synthetic cells, of course, but I think there is some relevance to it, and I hope that will become clear during the talk. Because I tend to forget thanking my people in the end, I wanted to just show this slide right in the beginning. So I have a group with working kind of all over the place, people from biophysics, biochemistry, chemistry, and so on. And the work I'm going to show today is mainly by former PhD students, actually Lucas and Aurore, who left the group already, and also Matthäus left the group. But Anna is a PhD student at the moment, and also Florian. They contributed something to the slides I'm going to show today, and also Andrea Mückel. She's a biotechnologist doing a lot of our cloning work in the lab. So Aurore Dupin, who was on that previous slide, she worked for many years on systems like they are shown here. These are the so-called droplet interface bilayer kind of networks, which have been studied by other people before, in particular by Hagen Bailey, which is an interesting system to study kind of simple, let's say, multicellular systems. And the idea here is that this is a water in oil bath, and there is lipid dissolved in the oil, and that actually creates a lipid monolayer around the aqueous kind of phase. And if you bring these droplets together by some mechanism, they actually form a double layer between them, and that essentially creates such systems where you have like aqueous compartments separated by lipid bilayers. So we were studying simple forms of communication between these systems for a while, and what you can also do is you can incorporate protein pores, like it's shown here, like the standard alpha hemolysin pore, and then let things permeate from one compartment to the other, and potentially also sort of switch on or off chemical processes depending on what you have in these droplets. So Aurore actually studied kind of different systems and different molecules to investigate such types of communication. And essentially, these are always kind of small molecules that are supposed to act as a genetic inducer, for example, in the end. And depending on the type of molecule, we actually put all kinds of different things into the droplets. So they could be either just simple in vitro transcription reactions, which we also like to call genelets, But in some cases, when we want to have protein expression, we also work with cell-free systems or cell extract. But in some cases, it was also useful to actually incorporate bacteria into the droplets. And in these experiments, she investigated whether you actually need pores in the bilayers or not for communication. And it's kind of relatively, well, in the end, it's kind of not super surprising what is happening, that it depends on how big your molecules are and how charged they are, whether they actually, or whether they are amphiphilic or not, whether they go through the membrane just like that, or they require a pore. And you see here are a few examples where you actually need alpha hemolysin to exert some kind of effect downstream. And the color code here just shows you that, for example, in this case, we used guanine in one droplet, and the guanine goes through the alpha hemolysin. And what we have here is actually a it's a fluorescent aptamer that actually is fused to a guanine aptamer, and that actually detects the presence of guanine. And so the green molecules are DFHBI, and you see that you can just detect whether the guanine has entered the kind of receiver droplet. And here you see that, well, this only works when you have alpha hemolysin present. But in other cases, and this is, the green molecules here, these are genetic inducers where we actually had E. coli present with the corresponding genes that coded for some fluorescent protein. In these cases, on the lower, on this row, it turned out that they actually don't need the alpha hemolysin. So DFHBI, of course, is actually the fluorophore that you use for fluorescent aptamers like spinach or broccoli. This actually doesn't require a pore, so it just goes through the membrane. And depending on sort of what our setting later on, we then had to incorporate alpha hemolysin or not. What actually also does not require alpha hemolysin is IPDG, also well-known inducer, and also tetracycline, and or ATC. So this is going to become important later on. So here's kind of a simple example of such a system. And in this case, actually what you see is a collection of, these water and oil droplets brought together with a micro manipulator. So it's really manually assembled. And what we have here is a sender droplet that contains guanine. And then we have these sort of guanine detecting kind of aptamers in the droplets. But what we also have is alpha hemolysin only in the red labeled cells, which means that alpha hemolysin incorporates into this bilayer, that bilayer, that bilayer. So all of the bilayers that are at the boundary or the border of the red arrow. So what then happens, is that guanine will diffuse into the system, and then actually, because there are alpha hemolysin pores in all of these bilayers, they will also diffuse into the neighbors, and therefore we detect fluorescence sort of in all the droplets defined by the arrow and the neighbors, but not in the further remote droplets. So that's a very simple experiment. But then, because these experiments were very cumbersome, so we actually to assemble essentially with the micro manipulator, we started to think about whether we can make bigger structures with 3D printing, and this turned out to be complicated. So we actually teamed up with a group at another university in Munich by Hauke Clausen Schaumann, who built actually a printer for us, and we have not really managed to make really big systems at the moment. So this is essentially what we get here. At least like this picture already looks a little organic, if you wish. But in the context of that project, and this is actually maybe the first like real part of my talk, we became interested in studying something like artificial developmental processes in such systems. So we thought it would be great if we said there is a gene circuit inside of these multicellular systems, and the circuit differentiates this multicellular systems into different parts, and that is of course motivated by, like, a standard example from developmental biology, the Drosophila development, and probably all of you know more or less how that works. So in the very beginning of the development of Drosophila, there is this there's an mRNA molecule deposited at one of the poles of the embryo. That's the becoid mRNA, and this is actually it's actually attached to the pole, and it's translated at that point, and that translation of course generates the becoid protein, which is then generating a gradient of the becoid protein inside of the embryo. And this gradient is then actually used by some downstream gene circuitry to determine where is the sort of the front and the back of the embryo, and then there is this kind of further definition of different regions in the embryo, like the stripe formation and so on. So we thought, well, it would be interesting to have something like that for a synthetic system, because in the future maybe we would like to create systems composed of synthetic cells that are supposed to somehow in a sense differentiate themselves in response to some environmental signal, and rather than making or generating this really complicated or using this complicated network from Drosophila, we used a much simpler network and put them in one of these kind of multicellular systems created from these droplet interface bilayer droplets. So here are three different topologies, or well, they are actually all very similar, but sorry, three different circuits that we investigated. And so essentially these are, we used repressors, genetic repressors like TET-R and LUC-I, fused to fluorescent proteins, YFP and RFP. So it's just false colored here. So the YFP is shown in blue. And the idea is that in the presence of TET-R, we would TET-R would repress the production of LUC-I and vice versa. So it's just a kind of a bistable or a mutually repressing arrangement of these repressors. And we just use IPTG, the inducer that acts on LUC-I as a morphogen. So rather than having a real kind of morphogen like in developmental biology, we just use IPTG. And we put that IPTG into our morphogen source droplet. And that means that because IPTG leads to a de-repression by LUC-I, that means in the presence of IPTG, we would expect to have a high signal of high expression of TET-R and therefore a low expression of the RFP. So that means that we would expect that in the neighborhood of that droplet, we should have a blue color in this color coding. And when there is a low concentration of IPTG, then actually LUC-I would repress TET-R, and therefore TET-R would not repress LUC-I, and we have an RFP signal. So remote from the morphogen source, we would expect a red color. So this is essentially it. And we did different kind of experiments here. One where we had a degradation tag on the LUC-I that's indicated here. So this is actually degraded in the system. And also we had another example here where we don't have the kind of this feedback where we only have repression by LUC-I. So when you perform this experiment, with this topology or this topology, then indeed you get something like what you expect. So you have a blue droplet here next to the morphogen source, and you get red droplets here and something intermediate here. So that was not unexpected, but the real question that we wanted to ask in the context of that work was not really whether this circuit works at all, but it is more motivated by a similar question that has been asked in the context of developmental biology, which regards the robustness of that kind of pattern formation. Because in developmental biology, the question is why or how do embryos, how is it achieved that embryos can differentiate so robustly in the presence of environmental noise? So every time such an embryo is generated, it will have kind of slightly different boundary conditions and things will change. But of course, because it's an extremely critical process for development, this has to take place very robustly. And here's a question regarding the size of the droplet. I will come back to that later. Now the question is, let's say in the future, we want to generate like synthetic cell-based materials, and they are supposed to also differentiate with respect to the environment. Then we will have a similar question. So whether this system would always robustly determine for example, position of a droplet correctly in such a gradient. And in the ideal case, you would like to have the first droplet should know I'm the first droplet, the next one should say I'm the second droplet, and so on. They should essentially know which number of which droplet they are in this network. And that's actually complicated. And this can be actually addressed with a specific measure that people have developed in this field, and that's called positional information. So you can actually put a number on the ability of a droplet or of a system to determine its own position from a gene expression level. And this is what we wanted to do in this work. So when you then perform these experiments again and again, so you make 100 of these systems, then every time you will have a different fluorescence level in these droplets, and this will create some uncertainty in your knowledge about the position, and this can be measured. And before I go to that, I wanted to just show you one video how that works. And there is already some variability in the system, so we actually try to be very careful to not to have an osmotic balance and to not so that the droplets are not changing too much, but it's very challenging to do this over several hours. We always have some slight rearrangement and movement of droplets, and there is a size change of the droplets over time. It turned out that this did not seem to be a major effect for the final result. But I'm going to come to this later. So formally, what is done, that's maybe not so, but only for the people who are interested. Essentially, the idea is that you have two variables. So you have a position X and you have a gene expression level G. And in principle, what you measure is the gene expression level that's shown here. And this has some variability from system to system, and from that variability you want to essentially determine what is my uncertainty in determining the position. So that is in a sense measured with this conditional probability. So the probability to be at position X given a certain gene expression level G, and obviously when you have a very broad distribution of gene expression levels, then you simply don't know which position you are at. And this can be quantified, and this is not maybe so important for you, but it's it gives you a number in terms of bits. So so this information in bits tells you that you can distinguish a certain number of regions within the system. So for example, if you have 1 bit, you can distinguish 2 to the power of I different regions. If you have 2 bits, you can distinguish 4 regions, 2 to the power of 4. And in our case, we have 5 droplets. So ideally, we would like to have in positional information more than 2 to be able to distinguish 5 droplets. So so when we did these experiments, so many times we get indeed a variability in gene expression level. So here's the droplet number 1, and this is the expression level. Sorry, the expression of this YFP going up over time, and the same for the second droplet, third droplet. So this becomes lower because the morphogen gradient, the morphogen concentration becomes smaller from left to right. And then from doing these experiments again and again, we get this variability in the gene expression level. And already from that, you can just, without doing this calculation that I was just talking about, you see that because of that variability here, essentially you don't know when you have a protein expression level in this region, you simply cannot know whether you are in droplet number 3, 4, and 5. So this is because they just have they are too variable in these regions. But then using kind of this mathematical expression, you can from these curves calculate the positional information, and then you see that actually this goes up. And then in the best case, we get like a little more than 1. So our system, even though it looks kind of pretty good in these images here it looks like you could actually distinguish 3 regions. But the point is that when you do many experiments, then in reality, you can only distinguish maybe 2 regions, which in the end is just, are you directly close to the, directly adjacent to the morphogen source or not? So that's a little disappointing, but I think that that's kind of was the goal, to actually figure that out, how robustly that is. What you also see here is that we can change all kinds of variables. So here is, for example, we changed the concentration of IPTG in the droplet, and what is and you see that there is that the system behaves differently well depending on the concentration of the droplet. And of course, if you have excessive concentrations like 100 millimolar, that essentially means that your whole system is flooded with the morphogen very quickly, and that means that you very quickly have concentrations above the induction threshold for LAC-I, and therefore you that means that you cannot distinguish anything in these systems. So therefore, when there is too much morphogen, then you have almost 0 positional information, because the expression level will be the same everywhere. And so there is an optimum in between. And we also find that we have generally more information in the blue or in the YFP channel than in the other system. Very good question. What protein expression system do you use? We use bacterial lysate in this case. It's a self-made lysate. So here's kind of a summary what we believe what happens. So one of the problems in the end is that so we have a certain induction threshold that's shown here. So we know that we need to have IPTG in this region to have a differential gene expression response. So if it's very low, then we don't see anything. If it's too much, then kind of there will be the same concentration or protein expression in every droplet. So we would like to be somewhere in this kind of sensitive region of that curve. And now then it depends on sort of in which whether our gradient that we generate sort of samples this sensitive region during the time that the system evolves, and this is kind of indicated here. So in order to get some differentiation, we would like to have so that's indicated by the gray bar. We would like to see that our the morphogen sort of concentration is within this kind of time window in this sensitive region. So as I said, when there is too much IPDG in the center droplet, then we get too high expression levels too early, so we cannot do any kind of differentiation. And now another thing, and that comes in, and that's kind of unfortunate with these systems. These are closed systems, so we incorporate the cell extract in our droplets, but these are closed droplets. So these are water in oil droplets. There is no connection to the outside. And in these droplets, simply the cell extract activity decays exponentially over time. And now one of the reasons why we have such a low positional information, and also why there is not so much information in this red channel, is that simply, and I'm sorry, I have to go back here, that in the lifetime of our cell extract, we believe that there is simply not enough time for this feedback loop to complete. Because what you would like to have is that the IPDG sort of diffuses into this array, and so therefore, like here, right in the close to the morphogen source, we would have a high TET-R expression, and you would have a kind of low, a high LAC-I expression further away, but then you would also like the YFP to repress the production of the RFP, but that takes some time. So this needs to repress this, and during the sort of because the system evolves over several hours, actually there is not enough time to complete kind of one of these feedback loops. So we cannot, and therefore we do not get these kind of intermediate expression levels that you would like to have to be able to distinguish several regions. So that would require a longer running cell extract or cell free expression, or some expression under some kind of steady state, but that's not the case in our case. And we think that this is one of the main reasons why the system doesn't behave so well. I see that I'm talking too long, so I might just switch to the next part. And the point, I think that in the okay, that maybe I didn't answer one of the questions. So we actually checked the sources of the variability that we see in these experiments, and we did some numerical modeling, and it turned out that the overall size of the droplets did not seem to matter too much, but it's really variability in gene expression in the different droplets, which we think is that we simply have different expression levels in each droplet, which might come from the compartmentalization in the first place. So it's not, so that was the main kind of influence in the end. But we have some variability in the sizes, as you see in these images, but they did not explain essentially the behavior. But sort of being aware of this problem that we have a problem with the finite lifetime of our systems, we actually in the last 2 years or so worked more with microfluidics to try to keep the systems out of equilibrium. And I'm just showing a few examples. So here's an oscillator that we worked on many years ago that's only based on transcription reactions, also compartmentalized in droplets. And here you see that it's oscillating, but then it's dying out, and you don't have a long-running oscillation. So that's a cycling video. Also, simply because all of the contents, the nutrients sort of are sort of exhausted, and the system just stops at one point. It decays to equilibrium. Of course, people have thought about this problem, and so there are solutions to keep the systems out of equilibrium for an extended time. And that here's one example by Roy Barziv's lab. You might know, and he actually immobilizes DNA on a surface in a microfluidic reactor, and then you can actually flush through cell extract all the time, and therefore the system is out of equilibrium all the time. So you do not have a decay of your cell extract because you replenish it all the time. And here they could actually show kind of extended oscillations in this system, so that the gene circuit is essentially immobilized on the chip, and you produce RNA and proteins from that, and they and this can be kind of run in a kind of a non-equilibrium steady state. In a different work, actually, Sebastian Merkel together with Enrique Niederholtmeier here, they used one of these systems where you have a kind of a ring reactor that can be separated from the outside via these microfluidic valves, and then you can actually inject stuff into this kind of ring reactor, let it react for some time, and then actually get rid of waste products and refill it with fresh reagents. And with this, you can actually perform also gene expression reactions under quasi-steady state, because you sort of always get rid of a little of the waste, and that can simulate, in a sense, they show that you can simulate something like cell growth, because also when, let's say, bacteria grow, you always have some dilution of your the contents of the cell simply by cell growth, but then also the cell has a metabolism, so it just generates new stuff for the daughter cells, and this can be more or less simulated in such a system. And with this, they also were able to show extended oscillations. And there is also work by Wilhelm Huck and Tom de Greve, who used the same system actually to study oscillations in this relatively simple system with an activator and a repressor. So I'm showing you these things because essentially these two things we also used in our work. We just also made one of these reactors, actually with the help of Sebastian Merkel, and we used one of these systems to study oscillations under non-equilibrium steady state. I still have questions on the previous part of the lecture. Maybe I'm going to maybe I can go back to that later. So first of all, alpha hemolysin was not required in these experiments because we actually used the IPDG as a morphogen, and that was the one that turned out to not require any pause. So this is really without pause. It just goes through the lipid bilayer. And sorry to go back and forth. And the other question is, can you go beyond 5 vesicles droplets? Or no, we actually in the paper we also had longer chains up to 9 or 11 or something, but it didn't improve. So we always just have a distinction between first droplet and the rest. So that is, yeah. And this is why I'm showing you kind of this part, that we think that in this closed compartment setting with cell extract, we will never be able to make any interesting dynamics, at least not with this approach, unless somebody invents some metabolic kind of metabolism, and then you can kind of also exchange chemicals with the environment. So here, what you see is a simple oscillator that is based on a sigma factor and TET-R. So this kind of gene here codes for sigma 28. And this sigma factor, together with the E. coli polymerase, kind of transcribes from this promoter here, and you produce a repressor, TET-R, and TET-R represses expression from this promoter. So this is a negative feedback cycle, and which is supposed to oscillate. And we just characterized kind of this oscillator motif in one of these reactors. I just wanted to show you a video how that actually works. So there is a ring reactor here, and we have valves that connect to this ring, and you see the red colored, that's just for visualization, the red fluid is injected into the into this ring, and then we use other kind of valves here to pump kind of the injected kind of fluid to mix everything. And what you saw in this image is that we injected only very little red fluid on the bottom and a lot on the top. And with this, you can sort of change, in a sense, the exchange or dilution per cycle. So you can decide whether you want to only replace a very small fraction or a large fraction of the contents of the ring. And with this, you can essentially control the dilution rate. And the dilution rate is an important factor in such a system, because the dilution rate essentially emulates or simulates growth of a system and or degradation of your protein products. And in the context of an oscillator, that is important, because the oscillations depend on the time of production of your protein contents, and also the degradation. So we have this kind of production of proteins by the cell extract, but we control the degradation or the dilution of the components using this feedback cycle, this pump cycle. And with that, we can actually then screen the parameter region. What you see here is this is the refresh ratio, which is actually controlled by the amount of fluid we exchange per cycle. And the transcription rate here, alpha, is something that we can not kind of control so well, but it can of course be tuned with also with inducer, for example. And here you see different examples of our oscillator in different parameter regions. So here you see up here there's only a low refresh ratio, and if we increase the refresh ratio, we get faster and faster oscillations. But you can also change, for example, the transcription rate, and that also changes the overall behavior, and everything can be modeled more or less well with a relatively simple ODE model. So these curves here are the models. So what we wanted to do here is actually studying whether we can use a microfluidic system to drive the oscillator. So much in the spirit of an electrical engineer, you would like to study whether there is some maybe resonance phenomenon. So you change an external frequency and see how your system responds to it. And to this end, we now used our feed cycles to also inject either ATC, which is the inducer, or TET-R as a repressor in certain periods to our into our system. And with this, we can try to actually somehow entrain the oscillator. So every time we essentially, we now give this biochemical oscillator, we kick it at regular intervals. And this is indicated here. So here we add the ATC, and this decay now is governed by the dilution. So it's an exponential decay over some time, then we kick it again. And here you see how these oscillations are actually influenced by this external driving. Now, as we saw that this sort of works, that we can actually influence the oscillator with our external kind of this external rhythm, we thought it would be cool to have some of interesting nonlinear dynamics phenomenon. And maybe you know from nonlinear dynamics that when you have a nonlinear oscillator, and this is one of the simplest ones, that's called the Duffing oscillator. So when you look at this equation, that's essentially a damped harmonic oscillator plus a cubic term. So that is only the kind of normal oscillator you know from mechanics plus a cubic term, and that's a driving term. And gamma is in a sense the strength of the driving. And when you do this, and this I just copied from Wikipedia, with this equation, then you see, well, first you get like these normal oscillations, then you increase this gamma, that's this term here, and then you see this pattern emerging, and this is what is known as period doubling. So you suddenly have two distinct rhythms in the system, and you can increase gamma more and more, and that actually also leads to chaotic behavior at one point. So people have actually studied similar behaviors in the context of circadian rhythms, and maybe you know that of course we have these day-night cycles, and people have kind of elucidated the structure of these networks, and they are essentially also networks, transcription networks that have positive and negative sort of feedback. I'm not going into the details here. So here's one paper that actually investigated numerical models of that, and also in this case you can actually see period doubling at one point. That's indicated here. So here would be a normal cycle, and here you suddenly have two cycles, and you can also get that become chaotic at one point. The point is that the issue is that this has never been really very well observed in the experiment, because in biology you apparently you need to, in order to show period doubling and chaos, you need to have very good long-term observations, and that seems to be very difficult in biological systems. And we thought, well, maybe we can do this with our cell-free system, and that's essentially the message of that paper, is that at certain external driving strengths, we now indeed see period doubling. So here you see that, for example, we have a big peak, then, or let's say we have, well, the first one is always the biggest, but then let's focus on this one. So first we have this red peak, then it goes down, then we have an intermediate, goes down, then we have a big one again. So we have kind of these different rhythms, in a sense, in this system, and that in this case it even indicates a 4 cycle, which means that there are 2 period doublings. So the system runs around. It's difficult to see. It first runs here, and then it makes kind of this cycle, and then it goes like this here, and then it goes to big cycle again. So this is an indication of period doubling in such a cell-free oscillator, which has not been observed so far as far as we know. And now we can actually use our exchange or refresh ratio or dilution rate to also as a bifurcation parameter. So this is actually, I think that's a modeling result, and it indicates that at one point we would have a 2 cycle or period doubling or a 4 cycle. And this sort of corresponds well to what we observe in the experiment. So here we have so this curve here corresponds to this parameter setting, and this looks like period doubling, and well, not exactly. And this is the example from the previous slide here. So we are actually quite excited that that works. And now maybe for the last two minutes or so, I wanted to just come back to this pattern formation thing. So because we have this issue with being in these closed compartments for pattern formation, we thought, well, maybe we can implement some type of stripe pattern in a microfluidics context. But this is really cheating in a sense, because we now actually, we don't really have a spatially extended system, but we just have different ring reactors here. And we feed these reactors different concentrations of the morphogen to, in a sense, simulate the morphogen formation, morphogen gradient. But in any case, what that allows us to do is that we can operate the corresponding gene circuit in a non-equilibrium steady state at these different inducer concentrations. And here's a incoherent feed-forward loop, which is also something that is sometimes used in the context of pattern formation. And the idea is that, for example, TET-R here represses kind of this M scarlet, but also there is kind of this other connection from TET-R to sorry, I'm confusing now. So here's another, we have a double repression here that is supposed to do some to result in an activation of the M scarlet at an intermediate concentration. So what is going on here essentially is that when we activate the node number 1 strongly, then that actually deactivates the production of TET-R here, and therefore we would expect to have an expression of this M scarlet at intermediate kind of levels here, whereas when we have a lower expression of this LUC-I here, then we would switch on TET-R, and that would then repress the M scarlet. So that in the end, if we perform this is the result of a numerical simulation, we would expect that we have a high kind of blue signal for the high input concentration here. You have a high M scarlet concentration for intermediate levels, and we have a high signal of M venous for very low signals of the repressor of the inducer. And now the only point that I wanted to make here is the following. So when we put this system with the corresponding plasmids into a cell extract in just a reaction flask or vial, then double repression, which is supposed to actually act like in a sense, activation of M scarlet doesn't lead to anything. So, but this system only works when there is actually a functioning double repression here. And this has to do with the fact that in the lifetime of a cell extract, there is not enough time for doing the double repression, because first you need to repress this, and then after a while, this protein first this protein has to repress this, and then this has to repress this, and then the concentration of that has to be reduced again. And this doesn't work in a closed reactor. But in this case, we put that into our kind of ring reactors. And here we see that actually double repression really leads to the desired activation. And in such a case, and now this is showing experimental results compared to the numerical results, and then indeed we get in the reactors that correspond to the intermediate morphogen concentration, we see an enhanced expression level. This shows that also this type of kind of stripe formation would work in principle if we had these kind of out of equilibrium conditions over extended periods of time. And here you also see when you monitor our reactor, for example, here that's reactor 4, I guess, over time, then you see that there's an initial phase where the behavior simply is not the one that we would desire for this kind of pattern forming circuit. But after, let's say, 5 hours, this initial transient is over and we settle to a steady state non-equilibrium state. And then this shows the desired behavior where we have a high M-scarlet concentration and low concentrations of the other proteins. And now you have to consider that when you work in a closed compartment, at this time, the cell extract will already be dead. And therefore, you will never be able to observe that behavior. So that's essentially kind of my talk, that where we studied kind of different types of pattern formation and or oscillations. And the point is that I think that one can do these, can make these circuits, and you can actually generate interesting behaviors, but, and this relates to the synthetic cell effort, that these things simply don't work when you operate them in a closed cell-like reactor. And so we only got them to work when we performed sort of these experiments in these microfluidic open reactors. And of course, that in a sense, for the whole kind of effort of building a synthetic cell, that poses this challenge that we somehow need to establish a metabolism and need to establish all these things that, of course, kind of real cells already have. And as long as we don't have that, we sort of need to kind of supply the metabolism and all these processes kind of externally with these means. And here I would like to say that these systems are really complicated. So here, and I think that there are other groups working with much more complicated microfluidic systems, but here that's our kind of reactor. So we have all this plumbing going on. And then inside we have kind of this chip and like all these things. And now you have to imagine, well, our cell-free system now sits in the middle of this, and all of these kind of supply lines are actually required to keep it alive in a sense, or out of equilibrium, to perform these functions. And I think that I recently talked to Michael Heimann, a colleague at Uni Stuttgart, and he made this comparison to that this is like an intensive care unit for the synthetic cell. So this looks like this. So essentially we have all these instruments to keep this cell-free system barely out of equilibrium or alive. And I think that's a big challenge. And I currently don't have a solution for it. And with that, I'm at the end of my talk. Maybe I still have a few questions in the chat. I'm sorry. Okay, thank you very much that it's not cheating. Thanks. And is there any fundamental limit to the kind of dynamic behaviors implemented by gene circuits? Oh, that's a big question. Well, I have maybe one, maybe not very good answer, but there is work by maybe you know Eric Winfrey and David Soloveitchik, for example. They worked on DNA computing and DNA and chemical reaction networks. And what they can prove is, in principle, that you can use one. They have thought about the computational power of gene networks or such networks, and you can prove that in principle you can simulate one system by the other, and in principle implement any function that you want in theory, and that, but on a practical level, of course, I think that one is limited by the number of how many connections you can actually implement between different kind of genes. So let's say you want to create larger and larger networks, then of course you need to, in a chemical context, you always need to be able to specifically address the genes, or which means that you need unique binding sites, for example. And at one point there will be, I guess, crosstalk that will be a major problem for scaling up these systems. And of course, if such chemical systems become larger and larger, then you run into many other problems, like also, let's say, simply time scales, and for let's say diffusion, for example. That was a big problem in DNA computing in the past, that when you wanted to scale up DNA computing to bigger sizes, you found out that there will be more longer and longer times required for diffusion of one component to the other, and that so therefore I would say there are fundamental limits, yes, but maybe it's not required to make so my question, and sorry, my answer maybe is a little complicated, is that the question is, what do you mean by fundamental in the end? I think that there will be usually always some limitations by simply the numbers of molecules that you can actually operate in parallel. I'm sorry. There's another question regarding the position information part of the target. We thought about position sensing circuits that modulate feedback on boundary permeability. I'm not exactly sure whether I understand the question, but I think it's an interesting question to maybe control the permeability with the gene circuits itself. So actually we have done that in one of the papers earlier on, that there was alpha hemolysin expression in response to an inducer, with the idea that actually this is kind of similar like in the luck operon, if you want, so that there is some let's say in the luck operon, lactose is diffusing into the cell, then it produces permease, which then allows more lactose to enter the cell. So we did something like that to generate some type of feedback, and I think that's a good idea, because it might help you to, for example, switch on or off diffusion in addition to get a better control over these systems. But at least for us, it did not work so well, but I think it's a good idea. Yeah. The reason why it didn't work so well for us, to mention that, is that we have in parallel diffusion through the membrane and through a pore. So there's always like parallel diffusion of non-specific and specifically gone through the pore. And one would need to probably think about the properties of the membranes and the pores very well to do that. Sorry, I took too long, Kate. I'm talking.
No, you're not. This is fascinating. I'm really interested in seeing that grow. Basically, do you think you would have better, I don't know, better reproducibility or less variability if you used something like pure system?
yeah, maybe.
Obviously,
actually,
you would go bankrupt.
Yes. So one of the reasons why we don't use pure for such experiments, and also the microfluidic stuff was not done with pure, but with cell extract, simply because it's a relatively large volume that you need. I still don't really know where the variability in gene expression really comes from. I don't know. So in earlier times, when we did these water and oil emulsion droplets, it seemed that simply the compartmentalization, like during the production process, we somehow affect maybe enzyme activities. Maybe there's some denaturation at the interfaces, and I don't know. So because when you do the math, in a sense, so these are very large droplets. These are hundreds of micrometers large. So these are not stochastic effects. It's not that you have stochastic gene expression, because we don't have just a couple of molecules. These are thousands or hundreds of thousands of molecules. So the variability comes not from this, but from I don't know what. So it's kind of strange. Maybe we just, yeah, I don't know. It could be that, let's say we have 1,000 components that are important, or maybe 100 in these cell extracts, and of every component we have like slightly less or more, and maybe a little is degraded or denatured, and maybe in total this generates such a big variability. Maybe, I don't know. It would be good to find out.
Yeah, that would really help, especially if we want to build like 200-300 gene circuits too. We have to figure that out.
Yes.
Okay.
Thank you very much.
Thank you so much. If there are no more questions, I don't see any other questions that we didn't answer. If there are no more questions, thank you so much again, and thanks everyone else.
Okay, thank you.
Have a great rest of your day and have a great week. Bye.
You too. Bye.